A rat picks up the scent of cat urine drifting across the floor of a lab enclosure. Every rat before it in evolutionary history has done the same thing on cue, freezing, then bolting for cover. This one does neither. It turns toward the smell, moves closer, and lingers there the way it would linger near another rat during mating season. Nothing is wrong with its nose. Nothing is wrong with its legs. What changed sits deeper, in the exact patch of brain tissue responsible for deciding whether a smell means danger or desire. A single-celled parasite did that, without touching a muscle, without releasing a drop of venom, without any direct contact with the predator at all. It reached into the part of the brain that assigns meaning to a scent and swapped one meaning for another. That is the part people miss when they picture parasites. The image that usually comes to mind is a worm eating through an intestine, or a tapeworm draining nutrients from a gut. Unpleasant, but fairly straightforward compared to what some of these organisms can do once they stop attacking the body and start attacking the decisions made inside it. Across 5 very different organisms, from a single cell to a virus barely larger than a strand of its own genetic code, the same idea keeps resurfacing. Take over the switch, and the muscles follow on their own. The rat sniffing at its own extinction is only the beginning. Toxoplasma gondii is a single-celled parasite, and by the standard of what counts as a body in the natural world, it barely has one. It has no muscles, no nervous system, no venom gland. What it has is a life cycle that only closes properly inside the gut of a cat, and every other species it passes through along the way exists only to get it there eventually. Rodents are the usual middle step, and this is where the manipulation becomes visible. Researchers exposing infected rats to the scent of cat urine expected to see the animals freeze or flee, the response wired into rodents for longer than cats and rodents have shared a name. Instead, infected rats approached the smell and lingered near it. Brain scans explained why. Activity in the posterodorsal region of the medial amygdala, the area tied to sexual attraction, rose to levels that matched what shows up when an uninfected rat encounters a potential mate. The same predator scent that should trigger the ventromedial hypothalamus's defensive circuit was instead lighting up the machinery for approach and interest. What makes this more than an accidental side effect is how narrow the change actually is. Infected rats still show ordinary caution around other threats, open spaces, sudden noise, the usual list of things a rodent is built to avoid. The rewiring targets cat odor specifically, not fear in general, which argues against random brain damage, and for something closer to a precise edit performed on one exact behavioral pathway. The parasite's own genome offers a clue to how. Toxoplasma carries a gene closely related to tyrosine hydroxylase, the enzyme that starts the chemical chain leading to dopamine, the molecule most associated with reward and motivation in mammal brains. A single-celled organism producing its own version of a reward chemical, timed to coincide with exposure to the exact smell that should send its host running, is not an accident of infection. It looks like a tool built for one purpose, closing the loop between predator and prey, just long enough for the cat to make the catch. That closing of the loop is also the entire reason the manipulation exists at all. Toxoplasma can multiply inside almost any warm-blooded animal, but it can only complete its sexual reproduction cycle inside a cat's intestine. Every other host, rats, birds, sheep, people, is a dead end, unless the parasite finds its way back into a feline gut before the current host happens to die of something else first. Rewiring one rodent's response to danger is not incidental cruelty. It is the shortest available route back to the one environment where the parasite can finish reproducing at all. The scale of this is easy to underestimate because the parasite itself is invisible without a microscope. Roughly a third of the human population carries a chronic toxoplasma infection, most without any symptoms worth noticing, since humans are a dead end for the parasite's reproductive cycle, rather than a stepping stone toward a cat. What happens inside a rodent's amygdala is not something a human brain necessarily repeats in the same way. But the existence of dopamine-manipulating machinery inside a parasite this common, already proven to rewire one specific fear into approach, raises a question researchers are still working through. About how much of what feels like a personal preference might occasionally have a passenger attached to it. A single organism editing one emotion is one kind of control. Another parasite living inside an insect, barely visible to the naked eye, manages something closer to remote piloting. And it does it while sharing the body with 2 or 3 identical copies of itself that never get to touch the controls at all. An ant infected with Dicrocelium dendriticum can carry as many as 3 of the flatworms at once, yet only one of them ever touches the brain. The rest sit in the ant's abdomen, encysted and inactive, technically along for the ride, but functionally doing nothing except waiting. The one that matters migrates to the subesophageal ganglion, the cluster of nerve tissue that functions as an ant's closest equivalent to a brainstem, and lodges there. From that position, it does not need to control every part of the ant's daily behavior. It needs to override just one decision, made once a day, at dusk. As the temperature drops in the evening, an infected ant abandons its colony's routine and climbs to the top of a blade of grass. Once there, it clamps its mandibles onto the tip and holds the position, exposed, motionless, waiting in a spot that puts it directly in the path of any grazing sheep or cow that happens to pass through the field overnight. This is the fluke's endgame. Its adult stage can only reproduce inside the gut of a grazing mammal, and an ant on the forest floor is nowhere near that outcome. An ant, frozen at eye level on a grass tip, ready to be swallowed with a mouthful of vegetation, is exactly where the parasite needs it to be. The timing carries its own logic. If dawn arrives and no grazer has come along, the compulsion lifts. The ant releases its bite, climbs down, and returns to ordinary foraging as though nothing happened, carrying on with colony work for the rest of the day. Heat appears to be the deciding factor, since rising daytime temperatures put both the ant and its unfed parasite at risk of drying out on an exposed blade of grass with no shelter nearby. Once evening falls again, the cycle restarts. The same ant climbing the same kind of grass tip, night after night, for as long as it takes. Confirming any of this took patience, rather than a single dramatic observation, since a successfully manipulated ant that finally gets eaten leaves nothing behind for a researcher to examine afterward. Much of what is known instead comes from following individual ants through repeated evening and morning cycles, marking which ones climbed, which clamped down, and which released their grip and walked away once the sun came up. Sometimes for weeks in a row before a grazing animal ever passed close enough to end the pattern. The backup copies waiting in the abdomen exist for one reason: insurance. If the controlling fluke inside the brain is somehow destroyed or fails, one of the reserves can, in principle, take its place and continue directing the same nightly climb. The parasite is not gambling its entire reproductive strategy on a single fragile connection between one worm and one nerve cluster. It is running the operation with redundancy built in, keeping backup pilots ready in case the one currently flying the ant goes down. None of this requires the fluke to rebuild the ant's body or take over its senses in any general way. Foraging, communicating with nestmates, defending the colony, all of that stays intact right up until dusk. The manipulation is scheduled, narrow, and reversible. A single compulsory action inserted into an otherwise unaltered daily routine. A different fungus living in the same forests skips the daily negotiation entirely. It does not ask its host to climb down again the next morning, because by the time it finishes, there is no next morning left. Ophiocordyceps unilateralis does not aim for a general outcome. It aims for a coordinate. Infected carpenter ants leave the canopy where their colony normally forages and descend to a narrow band low in the understory. And once there, Each one bites down on the main vein, running along the underside of a single leaf, at a specific height above the ground, in conditions of temperature and humidity that fall inside a narrow, repeatable range. Researchers studying these infections describe the resulting die-off sites as graveyards, because dozens of ant corpses locked onto leaves at almost the same height, in almost the same posture, tend to accumulate in the same patch of forest over time. The death itself arrives on a schedule. Field observations point to solar noon as the recurring moment when infected ants complete their final bite and stop moving. A level of temporal precision that is difficult to explain as coincidence across so many separate infections. For a long time, the assumption was that the fungus achieved all of this by taking over the ant's brain directly, piloting the body the way a hand operates a puppet. Closer examination told a stranger story. Fungal cells do spread through the ant's head, but the jaw muscles themselves show heavy atrophy and degeneration, consistent with the fungus manipulating the body mechanically, rather than issuing commands purely through the nervous system. The final bite may have less to do with a hijacked brain giving an order, and more to do with muscle tissue being restructured until clamping shut and staying shut becomes the only outcome left available to it. Whatever the exact mechanism, it has been running for a very long time. Paleontologists have identified death grip bite marks preserved in fossilized leaves roughly 48 million years old, a signature nearly identical to the one modern Ophiocordyceps produces today, meaning this fungal strategy predates most of the mammal lineages currently walking around it. The story does not end with the fungus reaching its target. Around a large share of these zombified corpses, a second, separate fungus moves in and colonizes the spore-producing structure the original fungus grew from the ant's head. This hyperparasite effectively eats the harvest before it can be released, and surveys of infected graveyards found that only a small fraction of spore bodies, well under 10 in every 100 by some counts, remained viable enough to actually infect a new ant. A patch of forest floor littered with zombified corpses looks like an active threat to any healthy colony nearby. Yet in practice, most of those corpses are already sterile, their spores consumed by something else entirely before they ever had a chance to spread. 3 layers of manipulation stack on top of each other in that single patch of leaves. A fungus controlling an ant's body, a second fungus quietly sabotaging the first, and none of it visible from more than a few steps away. Somewhere nearby, in the same damp leaf litter, an entirely different kind of parasite is preparing to drive its host toward water instead of a leaf, aiming not at one insect's death, but at feeding an entire population of fish downstream. A camel cricket infected with a nematomorph hairworm carries something that has no use for dry land. The adult worm can only mate and lay eggs in water, which puts it in the position of an animal that spends its entire reproductive future trapped inside a host built for exactly the wrong environment. Its solution is to change where the host wants to go. Infected crickets show an altered response to reflected light, moving toward the glare bouncing off a stream's surface far more often than uninfected individuals in the same field. Once a cricket reaches the water's edge, it does not simply stop. Field researchers in Japan have watched infected crickets walk directly into streams and continue moving until they are fully submerged, at which point the worm, sometimes several times the length of the cricket itself, exits through the body wall into the water it needed all along. A worm that size, emerging from an insect a fraction of its length, is not a subtle event. Some nematomorphs measure longer than the cricket's entire body once fully extended, packed inside the abdomen in tight coils for weeks until the moment of emergence finally unspools them into open water. The cricket rarely survives the process for long, and in the stream studied, a large share of the ones that make it into the water do not last long enough to matter anyway, because trout are waiting. A multi-year study across 9 separate streams measured what these fish were actually eating and found that terrestrial crickets made up close to half of trout stomach contents by mass on average. A striking figure for prey that, under normal circumstances, has little reason to enter the water voluntarily at all. The detail that turns this from an interesting coincidence into a documented ecological chain is what the researchers controlled for. Cricket consumption by trout tracked closely with how many nematomorph worms were present in a given stream, not with how many crickets happened to be living in the surrounding forest. Streams with abundant crickets nearby, but few worms, showed comparatively little cricket predation. Streams with heavy nematomorph infection showed the opposite, regardless of how many crickets were actually available on land. The parasite, not the prey population, was setting the terms of how much terrestrial biomass entered the aquatic food web that season. That reframes the hairworm's role inside the ecosystem entirely. It is not simply killing its host to reproduce. Every successful manipulation moves a package of nutrients that originated on land, built from leaves and other insects a cricket ate over its lifetime, directly into a river system that would otherwise depend on drifting insects and whatever washes downstream naturally. A single species compelling its host to drown has, indirectly, become one of the more reliable food subsidies certain trout populations depend on. None of the crickets driven into these streams die from a bite, a sting, or a physical struggle with anything. They die because a parasite living inside them rewired what looked attractive across a stretch of open ground, and the rest followed on its own. Something smaller still, without even a body of its own in the conventional sense, achieves an outcome just as total, using nothing but a single borrowed gene. A virus does not have muscles to hijack, or a nervous system to invade, since it barely qualifies as a living thing between infections. What certain baculoviruses have instead is a single gene, EGT, that produces an enzyme capable of disabling ecdysone, the hormone that triggers molting in caterpillars. Under normal conditions, a caterpillar's growth is interrupted repeatedly by molting, a process that can take it offline from feeding for roughly a full day at a time, while it sheds and rebuilds its exoskeleton. An infected caterpillar loses that interruption entirely. With ecdysone neutralized, molting never properly triggers, and the caterpillar keeps eating and growing without pause, manufacturing more biomass for the virus replicating inside it to consume. The same disruption bleeds into the caterpillar's daily schedule in a second way. Healthy caterpillars typically avoid daylight exposure, hiding in bark crevices or leaf litter, and feeding mainly after dark. Infected individuals abandon that pattern and climb toward the top of the plant, remaining exposed through daylight hours in a shift long known among foresters as treetop disease, decades before anyone identified the viral gene responsible for it. The payoff for the virus arrives at the caterpillar's death. Instead of dying hidden near the ground where a body would decompose largely in place, the infected caterpillar dies at the highest point it reached, then liquefies there. Viral particles released by that liquefaction rain down onto the leaves below, exactly where the next generation of caterpillars on that same plant will be feeding within days. A behavior that looks, from a distance, like a caterpillar simply exploring upward, turns out to be the delivery mechanism for the virus's entire next generation, engineered through nothing more than one gene interfering with one hormone. Return to the rat that walked toward the smell of a predator instead of away from it. That rat's amygdala had been repurposed, not damaged, redirected toward exactly the outcome its parasite required. The caterpillar climbing toward daylight it should be avoiding. The ant clamped to a blade of grass at dusk. The ant biting into a leaf vein at the exact height and hour a fungus requires. The cricket walking into water it has no reason to enter. All of them share the same underlying fact. None of these organisms lost a fight. None of them were overpowered by claws or venom or superior strength. Something far smaller than them simply found the one switch controlling behavior that mattered and turned it. The bodies stayed intact until the very end. It was never the body that belonged to the host anymore.